welcome to this three-part interview series where we'll be looking at bioanalysis for biosimilars. Today I'm joined by Stacey Gottschall, who is the group lead at QPS's Translational Medicine Department. So welcome, Stacey.
Thank you.
So my first question for you, Stacey, is I was wondering if you could give us a little bit of an overview of some of those major biologics that have recently gone off patent or are expected to lose that exclusivity in the next couple of years.
So the loss of patents of the biosimilars is really driving the boom right now. Some of the major biosimilars that have recently lost patents over the past few years includes Humira, Rituxan, Herceptin, Avastin, and Remicade, and more recently Stelara. And these represent well over $60 billion in peak annual global sales. And many of these biosimilars have already been approved. Looking through 2033, we're looking at the next wave of biosimilars that will lose patent, including Keytruda, Opdivo, Darzalex, which is expiring in 2026 through 2029, as well as Dupixent and Skyrizi. These represent a massive $55 billion. The impact of the biosimilar market is profound, and the accelerated development of these pipelines and more aggressive investment will help with manufacturing and analytics, and it'll drive the market to be more competitive, lower prices, and increase the availability for patients.
So my next question is then, how do you predict that these changes are going to impact the biosimilars market?
Among the biologics approaching this patent cliff are Keytruda and Opdivo, which are the biosimilars in development, and they're PD-1 inhibitors. Together, they're generating over $35 billion in global sales. They're not just first-line therapies, they're also across multiple cancers, and they are embedded in combination regimens as well. We should also highlight Darzalex as an anti-CD38 therapy, which is roughly $12 billion in sales. This composition of matter is expiring in 2026 with an additional decline in maturity in 2029. For biosimilar developers, the price across all these assets is clear in the market sharing. It'll increase competition. The complex monoclonal antibodies, where there's minor differences in the structure, it'll increase the safety, efficacy, and immunogenicity, giving the bioanalytical package, right, what turns a promising candidate into a regulatory success.
And then I'm interested, so as of 2025 and 2026, how many biosimilars have been approved by the FDA and then the EMA?
So for the FDA, there's approximately 82 biosimilars that have been approved through the 351(k) pathway.
Okay.
Some sources note up to 96, which includes some of the older biosimilars approved prior to the current pathway. In 2025 alone, the FDA approved a record 18 biosimilars. For EMA, the numbers are even higher. EMA has approved upwards to 165 biosimilars, with a record year in 2025 of 41 biosimilars. Some of the highest annual total to date, and some of the major ones include Humira, which the FDA has approved 10 biosimilars for. Stelara, which multiple biosimilars have been approved through the US and EU, and has given additional opportunities, options for psoriasis, Crohn's disease, and ulcerative colitis. Perjeta was notable in 2025, which was the first biosimilar to HER2. Prolia, which is a large wave of approvals. So there was 8 for the FDA and even more for EMA for osteoporosis and other cancer bone diseases. The oncology standouts include Avastin, Herceptin, and Rituxan. The surge in approvals is especially complex for the monoclonal antibodies and newer reference products. This is going to increase competition, improve access, and it'll decrease pricing and allow easier access for patients.
Can you tell us a little bit more about how the landscapes differ for biosimilars between the FDA and the EMA? And what guidance documents have maybe shaped those frameworks?
So the FDA and EMA both use a stepwise approach and a totality of evidence. So they're looking for the analytical characteristics. They're looking for to improve biosimilar to highly similar compounds. They're doing at a molecular level. Then moving into non-clinical studies, into clinical studies, and potentially clinical efficacy data. Both EMA got there first, so they're more experienced looking at a data-driven system. The first biosimilar for EMA was approved back in 2006 for the FDA. Their pathway started in 2009 and 2010, with the first biosimilar approved in 2015. In April of 2015, the FDA released its fundamental guidance to lay the stepwise approach and set clear expectations for what data sponsors needed to provide for biosimilars. Then in October of 2025, the FDA issued a major draft guidance to provide more clarity around the biosimilar and what data was needed and what the acceptance criteria for those were. They also talked about the need not to run the large efficacy studies similar to a large phase 3 clinical trial. Instead of running those trials, they're looking more at strong analytical data. They're looking at the in vitro pharmacology and the comparative PK studies between the biosimilar and the reference product. On the EMA side, back in March of 2013, the Biosimilar Medicine Products Working Party drafted the revision, and this was later adopted in 2014. This became the cornerstone of the biosimilar development in Europe, reinforcing the comparability exercise. In April of 2025, EMA released its draft for clinical approach in biosimilar development. It suggested that under the right conditions, biosimilars could potentially be approved with a reduced or even a waived comparative efficacy study with some high quality pharmacodynamic data, as well as the analytical comparability, in vitro data, and solid PK data.
So the latest FDA and EMA documents, they place a much greater emphasis really on strong analytical data. And that's via those comparative analytical assessments, so the CAA assessments, over the comparative phase 3 efficacy studies. Can you tell us a little bit about what factors drove that shift and how this is going to be significant for developers?
This represents a real shift in how biosimilars are developed and approved. For years, sponsors were using the expansive comparative studies, similar to the large phase 3 trials, and how well the biosimilar performed in the patient population. Now both the FDA and EMA are saying if your analytical data is truly rock solid and a comparative analytical assessment, those big efficacy trials aren't necessary. The major improvement is the advance in technology to characterize the complex proteins, looking at structure, purity, formulation, impurities, and biological activity, which increase sensitivity and specificity. Regulators have come to realize that these sophisticated lab-based methods will find any differences in your biosimilars or your reference product than compared to the large population trials. The second is the real world experience. So both EMA and FDA have reviewed many biosimilar studies with the first approvals in 2006. So both agencies have reviewed these programs, and the vast majority of the cases, the comparative efficacy studies are not changing the final outcome of whether the drug is approved. The practical and policy goals is another area to focus on. So the large efficacy trials are very demanding. Their cost is high. They take 1 to 3 years to complete. Drives an overall development to increase the speed of the development, making sure there's not delays in the market, and getting the biosimilars to the patients in faster and at a more cost effective. With the updates in 2025 and over a decade of experience with the analytical tools, we're seeing both agencies coming together and getting on the same page of whether those efficacy trials are needed. If you have the strong analytical data to show the similarities with your compound, as well as the comparative clinical studies, they may be wavering the need for some of the efficacy trials.
I was wondering if you could share some examples of biosimilars that QPS specifically has supported in development, and maybe among those molecules or indications that you've worked on specifically, which ones stand out for you in terms of their significance or maybe their associated challenges?
So QPS has supported 6 biosimilars: Avastin, Remicade, Rituxan, Herceptin, and Humira. We've also developed and validated robust bioanalytical methods to support several other key biologics, including Keytruda, Stelara, Darzalex, and cetuximab. One of the most memorable challenges was with the early rituximab program. We had the simultaneous approvals in both US and Europe. So we had to demonstrate analytical similarity between the EU approved reference product and the US approved reference product, which meant that we were comparing the 3 versions of the molecule side by side. So the biosimilar, as well as the 2 innovators. It significantly increased the complexity of both the method development and the validation with the workload. We had to build a careful, balanced study design to make sure that we were able to compare all three calibrator curves together, as well as all of the QCs on the same plate across different analysts, to ensure that they were still showing the similarity within the bioanalytical method. Another notable case involved developing 2 separate cell-based neutralizing antibody assays. At this time, it was still highly debatable whether a single method or 2 methods was the appropriate approach. And with the cell-based NAb assays, it's difficult to keep one going, so that the time constraints that go into keeping the cell-based assay ready and throughout the life cycle of the program, we had to make sure that we were maintaining 2 separate assays. And so thankfully, the field has really gone into looking at a single assay approach, which has really helped with some of the early work that was done and how much work was going into those methods.
So we're going to wrap it up there for part 1, but please join us for part 2, where we're going to be looking at method development and best practices for PK similarity studies.